we are very pleased to have our speaker, Mr. John Toomey. He actually operates a business out of the Mountain Business Park here, and I believe it's in not T-Building, but it's an operating support building.
Yeah, the Advanced Technology Center.
Advanced Technology building. That was built kind of after I left, or when I left, so I don't know that. But anyway, we're very pleased to have John here, and I want to thank everybody for showing up. And John, it's yours.
Hey, thank you very much, Bob. Appreciate it. And thank you, everybody, for being here tonight. Appreciate the support. So just jumping right into it, I've got a testing laboratory located in the Mountain Advanced Technology Center. We've been in business for about 3 years now, and I've got a little bit of a talk here today. It's going to go over really, the role of the testing laboratories in the medical device space, how it, relates to sustainability efforts medical device companies are currently involved in, as well as some safety and compliance items. And so just a little background about myself. So I went to Wright State University. I have an undergraduate and a graduate degree in chemistry. I studied synthetic organic chemistry and then kind of pursued a biological sciences PhD, but kind of got quickly drawn away from that and took a sabbatical to go on an entrepreneurial journey. And and this is where I've ended up now. I'm at Bioscension, right? But Bioscension was founded shortly after I started my entrepreneurial journey. The first venture was more in a health and wellness type setting. This is still kind of in a similar realm or field, but it is medical device, and I've always kind of had a passion for medical device, and I've watched my father. When I was growing up, he was an engineer for medical device companies, had several intellectual property and patents, and I got to watch him work and see what he went through, and I think I always kind of gravitated towards that, acting as an intern at some of the businesses that he worked for and started himself as well. So. Just a little bit about Bioscension. So I told you about our founding year, right? So we were founded in 2018, but then our accreditation status came about in 2022. And I'll talk a little bit more about the accreditation status the laboratory holds and some of the standards and regulations that the laboratory is governed by, right? Same goes for medical device companies as well. They're governed under a similar set of standards and regulations by certain regulatory bodies, something like the FDA. And USP strictly governs, some of the test protocols and procedures that we perform in the laboratory here at the Mountain. So we have a wide and a full range of testing. It's microbiological based, it's chemical based. So we do a lot of, it's microbiology where you're doing growth promotion testing. You're looking at antibiotic potency testing. Things of that nature. Chemical side of things, though, we do a lot of water testing, and that goes hand in hand with TOC as well. So we have a few analyzers that perform some TOC analysis on site. And really, we set out. So it's not just myself that founded the company. I actually founded this company with my brother. So family, kind of family ties, and also some other individuals that I met, over time while working in this space. And we started out as a QA/QC testing laboratory and, helping these medical device companies with their research and development programs mainly. And then we kind of evolved into, a full-on lot release as well as, regulatory and compliance submission-based laboratory. And going back into our focus, right? So the focus is we want to offer testing that's at a reasonable cost, that has fast turnaround time, and you also get meaningful customer service along with that. We're not just issuing reports. We're giving you information that can help you guide your medical device process, your manufacturing or your reprocessing process. And we primarily work with small and large medical device companies, as well as healthcare systems and healthcare facilities. And so I mentioned that we were accredited, and so we are ISO 17025 accredited laboratory. We obtained that accreditation status in 2022, and actually in about 7 days, we have another recertification audit coming up. So I've been, prepping heavily in the laboratory and staying busy and making sure, all the I's and the T's are dotted, and that mainly goes into, a lot of these items that you might see here. And so this is kind of let's say if you were a company and you were trying to obtain this accreditation status, these are the types of guidelines that the standard lays out that you would follow to obtain said accreditation status, and also offer testing and calibration services. And so ISO 17025 primarily governs testing and calibration service companies. And there's several items that go into this, and I just want to point out a few of these, such as documentation, training and awareness, process alignment, internal audits, management review. Pretty much anything from 4 all the way on to 10 is, and 10's the stage that we're in right now for the recertification portion of it. But these items go into basically a sound and robust quality management system that laboratories follow, so that you can ensure that the results are reliable, that they're credible, the test methods are validated, and obviously this insight's being offered to large individuals in the medical device organization, whether it's manufacturing, reprocessing, maybe pharmaceutical industry as well. And so having these things in place and these methodologies validated is very important to those individuals. So it's hard to you jump into business with these types of organizations without carrying an accreditation such as this. So I just want to jump into history of medical device and healthcare industries. So this is, I would say, a very bare bones historical timeline. So this definitely doesn't cover everything that's happened from the 1800s on to modern day. But I just wanted to touch base on some of the things that are primarily relevant to this talk. And the first thing being, here in the 1800s and the 1900s, we had surgical innovations begin. And we also had, the development of specialized forceps for certain medical procedures, as well as some implants, early implants. And moving on to mid-20th century was the disposable syringes were developed. And so the first disposable syringe was developed in mid-20th century, as well as, pacemakers kind of came onto the scene. And dialysis and joint replacement became, clinical operations or things that could be performed to improve patient outlook for individuals suffering from, certain disease, especially renal in this case, and then arthritic for the joint-based portion. In the 1970s, so I kind of find this being an interesting point. You have FDA really stepping in and kind of setting the bar for what they expect from manufacturers and anybody who's manufacturing medical devices, as well as pharmaceutical companies that are developing drugs. And so, and also around the same time, we had the CAT scan, so invention of CAT scan, MRI, and other biomaterials, so things that could be utilized, that they're more polymeric-based materials in, I would say, medical-based procedures. 1996, so ISO 13485. So I mentioned ISO 17025. That's for testing laboratories. 13485 is for medical device manufacturers. So medical device manufacturers following very similar guidelines, the corrective actions procedure, the documentation, validation of certain methods, that was standardized, and QMS was essentially developed in right around 1996. Excuse me, 1996. I'm going to call this AAMI, AAMI, USP, ASTM, and ISO. I have the breakdown of the acronyms on the next slide, but you get a feel for the types of standardization bodies that kind of govern these types of activities, not only for medical device manufacturers, but for testing laboratories as well. In 2020 to 2022, as you all know, COVID-19 pandemic hit. And one reason I want to bring this up is because there was a huge innovation surge, and this was for, immunological assays. Testing labs were absolutely filled to the brink with test articles and samples from mask, N95 mask manufacturers, looking for and evaluating the cleanliness of those masks and making sure that they were actually effective at protecting an individual from, said pathogen. And then digital health as well. So I feel like digital health was probably still early 2000s, maybe 2010 based activity. But with the advent of computers and then specialized software, we're able to improve patient outlook, and as well as track a lot of the things that medical device manufacturers as well as testing facilities are responsible for keeping track of. And present day, so 2000s, robotics, smart implants, AI diagnostics, as well as wearable devices. And so I actually brought a example of a robotic device. So this is from a Da Vinci robotic. It's basically a surgical robotic suite. And so this device has unique features and varying degrees of axes that can be controlled by a clinician inside of a surgery center in the hospital. And so this is actually an example of one of the devices that we work with, we provide testing on. And I'll talk to you a little bit later about the complications of testing items like this, because medical devices come in all different shapes and forms. Some might be tissue-based and not actually a device, a type of item or a test article. So that definitely makes our job a little bit more complex. And this is so just a review of the timeline again, but I wanted to just show some images here. So I thought this was interesting. The first hypodermic syringe. So Dr. Alexander Wood kind of developed this concept or this idea. So this is a very, I would say, rudimentary version of the syringes that we know today. Right now we're using polymers that have been sterilized by one way or another, or we have access to glass as well. So high quality borosilicate. So CT scan, MRI, ISO 13485. Here's the Da Vinci robotic surgical suite in use right here. So you can see that outside of this device or the extension of that surgical suite, there is a lot going on in the operating room, and you basically have a single individual, obviously a surgeon, right, operating this instrument, and there's certain instructions for use that they have to follow in order to perform these procedures and improve patient outlook. One last thing. So Dexcom, I don't know if you're familiar with Dexcom, but they do wearable glucose monitors. So that is, I was talking about wearable and types of devices and the integration of that technology in modern day. And this is definitely something where I think there's a lot of movement in this space, and it's not only for glucose monitoring, but they're working on other technologies as well. And so standardization, right? These are the governing bodies. These are the standard organizations. They commune internationally, and they determine what types of steps need to be taken to ensure that ultimately medical devices are safe when they reach a clinical setting. USP a little bit less so, because that's more of the pharmaceutical side of things, but just because it's pharmaceuticals and I'm primarily talking about medical devices doesn't mean that it wouldn't be something that a testing lab like Bioscension would be expected to handle. We certainly do have some pharmaceutical type test articles that come through as much as medical devices as well. FDA, so you're probably all familiar with FDA, so it's Food and Drug Administration, and then they should add an M for medical device as well, because they also govern medical devices. And that is, and one key thing, so if you're talking about different classifications of medical devices, especially class 2 and class 3, you'll have to file, all the companies, so manufacturers are responsible for filing a 510K with the FDA, and that is a rigorous process in which they review the device, the documentation behind that, the process that they use to manufacture it, and basically the entire step of, all the steps that are involved in manufacturing a device. ASTM, so just more as I would say is generalized for methodologies that you might employ in a test laboratory. So they have certain test standards and test methods, which basically govern how a test should be performed in order to be compliant with this particular standard. And AMES, I would say, falls in a similar boat, but it's definitely more towards, it's geared towards medicine. And so they develop a lot of standards that would maybe cover endotoxin, so endotoxins on medical devices.
What does that stand for? AAMI?
Yeah, so the American Society of, Association for the Advancement of Medical Instrumentation. And so this definitely, I think that kind of broadly describes what it is that they do, but they look a lot outside of just medical instrumentation, and they kind of govern, I would say, some pharmaceutical testing that's being done, as well as just the methodologies that are being performed when you're developing, because let's say you develop a device, you have to develop a test plan to accurately say what types of substrates or contaminants are on this device. And these guys govern exactly how that should be done. So they talk to you about, extraction parameters, which might be based on surface area. So, they say exactly, what that surface area is on a per centimeter squared basis that you should be extracting these devices, so that you can ensure that when you're getting ready to test them and you extract the devices, that you have adequately extracted the contaminants from the device and you're not leaving anything behind. So moving on from standards, so again, these, keep these in mind, because you'll see these kind of pop up again during this talk, and it's going to govern the test processes and the procedures that I'll talk about in the latter half of the seminar. And I want to move into the state of medtech in 2025. So I gave you the historical timeline, told you a little bit about the standardization of medical device manufacturing, medical device reprocessing, if you will. And I think this is a key thing to focus on, especially for testing laboratories, and it's very pertinent to the topic of this talk. And in 2025, and let's talk about 2024 first, but in 2024, we've seen significant growth in scaling, and we're talking about, in the US alone, there's 6,500 companies that are registered medtech firms that are developing, a medical device or an implantable or some form of product, right, that's going to improve patient outlook. And there's a constant evolving regulatory complexity. So again, going back to those standardization bodies, they're putting more rigorous requirements on these companies to ensure that these devices are safe when they do reach the clinical setting and when they're released from the manufacturer. Because if you don't have any of these controls in place, that's when people become ill from a procedure as opposed to being healed from it, essentially. And so just, again, I don't want to get too much into the numbers, but the industry is booming. And there's a lot going on. There's a lot of, I would say, support industries that are involved, such as sterilization and testing laboratories. And I would say both of those industries are being bogged down by the demand of these med tech companies here in the United States, but not only the United States, but in the EU and other regions as well, Asia, for example. So going into the process of medical device manufacturing and the reprocessing. And so I want to make a clear distinction here. And before I make this distinction, I want to talk about, how are these devices designed and what are those designs based off of? And then what process goes into designing those devices, and not only the devices, but the process that's used to manufacture them. And the tests that are required to ensure that they're safe. And so for making a medical device, right, you would have input raw materials and you would manufacture the device. So in this case, we have, some snips or some form of, suture snips. And then at this next step, you have testing. And you'll see testing is repeated again. So you have post-sterilization testing, and pre-sterilization testing. And that's actually in both cases. So for manufacturing the device from scratch and then from reprocessing a medical device. So the difference between these two is that medical devices, and if you're familiar with this, they are actually taken from, they're used, and then they're disposed of in a biohazard container most of the time. But as of recently, maybe early 2010, this became, a very popular thing. Individuals set out, they became regulated under the FDA, and they reprocess these devices for use. And so by reprocessing, I mean they might clean them, they might repair them to a state that is exactly like a newly manufactured device. And they go through all of these steps very similar to what you would experience if you were to just manufacture the device from scratch, from the raw materials and the other components. And in the design process, engineers are very much involved, and testing laboratories and sterilization facilities are working and collaborating with those individuals at the medtech organizations constantly. And so, going into the next slide here, I want to talk about the FDA classifications. So you made a device, right? Now, what classification is that device? What class is the device, and what does the classification mean when it comes to the regulations surrounding that device and the cleanliness of that device? And so class 1, you were thinking of analyzers. Another thing I think of is like crutches, wheelchairs, things of that nature, right? So these are items that are not necessarily going to be contacting a patient in a surgical sense or in a procedural sense, but they are something that is a medical or it's a medicine-based item, right? And it helps patient outlook. So anything that improves patient outlook is going to be classified by the FDA as either class 1, class 2, or class 3. Moving on to class 2 devices, we have the ventilators. So COVID, right? Ventilators popped up. That was a very frequent topic, as well as some surgical clamps. So if you're dealing with any surgeries where you break your leg or something like that, they need to go in there and clamp something together, put certain metal components in to hold or fix the bone into a certain orientation. Those would be those devices. Bone grafts. So again, I'm talking about devices, but then here's bone grafts. So this kind of pops up. So that's another interesting piece. But again, this is procedural based, but there's also certain, I would say, manufacturers, if you will, that are responsible for bone grafts, as well as skin grafts would fall under this category also. And I would say the primary device that we work with most is a testing laboratory, and that has the most critical and stringent requirements are your class 3 devices. And so these are your pacemakers or anything that's implanted, or maybe it's a blood contacting catheter, right? So an electrophysiology catheter, or anything that essentially is going to enter the body. A surgeon is implementing that technology to improve patient outlook. These would fall under class 3. And I want to touch base on medical device reprocessing. I already explained that it's kind of a new advent, and before 2010, individuals were just manufacturing new devices from the raw materials, and they weren't thinking about, okay, well, we could recycle these devices, we could collect them from the hospitals, we can bring them back to a reprocessing facility, and we can clean these items, and they can be deployed again into clinical use. And so in the 1980s and 1990s, they were very much single use. So you're dealing with single-use devices, and a lot of devices, even some of the ones that I have here on my left today, are deemed single-use devices, but are taken in from the hospitals and under a separate 510K are filed as a reusable device. But they have to prove that those devices function exactly as the original device that was manufactured functions, and that the cleanliness, right? So, and I'll tell you a little bit more about cleanliness. There's obviously biological contaminants, chemical contaminants that could be on a device, especially when we're talking about reprocessing, that they need to make sure are absent from that device before it is deployed and released to these clinical settings. And so where does reprocessing happen? I thought this was interesting, but so hospitals, obviously, or you think of your dental clinics, right? They're going to take certain things that they might use in dental procedures, which may be handheld or manual tools, but they will be reprocessed typically by autoclave sterilization and reused. And it gets a little bit more advanced when you talk about a catheter that has moving pieces, or it has a fixed curve, or maybe we're dealing with a transeptal needle, right? So these needles, they need to have a certain degree of straightness or hardness, rigidity, right? So when they're being used in another procedure. So I would say a lot easier to process certain devices on site at hospitals and clinics, and they have sterile processing departments that handle exactly that. Just imagine a room, it's a very hot room, by the way, filled with autoclaves, a wall of autoclaves where they're placing instrumentation in, and that's primarily the tools that they would use for those manual-based procedures. Now, Another interesting thing that hospitals and these clinics are doing is scope reprocessing. So they have specialized units. So they're called automated endoscope reprocessing units. And so they actually reprocess, wash, and clean the endoscopes and reuse them. And that is a very common practice in hospitals and clinics across the United States. And again, all of these activities are governed and regulated by FDA and some of the other standardization bodies that I mentioned earlier. Let's talk about the offsite. So offsite is where the devices are collected from the hospital. So the hospital says, hey, we can't reprocess this cardiac catheter. We don't even know where to begin. There's all these different components. It has special coatings. The handle's bent. The connector cable is damaged, right? How do I fix this? How do I repair this and make this into a device that could be reused again? And so that's where these off-site and centralized reprocessing facilities come into play. So they're specialized. They have groups of engineers, that I mentioned, that work in the design process, that basically develop procedures and reassemble or take, let's say, new raw materials and incorporate them into the device so that you're not throwing out the entire device. So electrophysiology catheters, a lot of the time they have titanium or some rare earth metal present in them. And for several years they were just discarded. And so being able to recapture especially those components is important for the sustainability, and also I think it helps address some of the demand and the need, because we're dealing with a lot of illness and disease. So we need to make sure that we have those devices available. If we don't have the necessary metals or the necessary components that are required to accomplish that procedure, then patients and individuals will suffer. So I think reprocessing is a very important piece of the medical device industry, and reprocessing, another word for that would be remanufacturing. So, but it's taking a manufactured device and it's making it usable again in the clinical setting. And third-party remanufacturers, I would say they work hand in hand with these off-site centralized reprocessing facilities, because let's face it, some of these engineers are not skilled at or understand the coatings that are on the catheters, so they send that off to another individual that will apply the coating or a specialized coating that will return the device to essentially as good as it was once it was initially manufactured. And one other key thing to be aware of is when devices are reprocessed, the packaging has to indicate as such. So you will see packaging from manufactured or newly manufactured devices which doesn't indicate anything about reprocessing, but anything that has gone through this cleaning process will indeed have some marking or some indication on the package that this was a reprocessed device. And I'd say the purpose and impact, right, is it saves money. It not only saves money, I would say, at a patient level. So, right, you get your medical bill after a procedure. It's typically a high amount, and they're using the newest devices for several years. So, of course, it's going to be expensive. And we're talking about the Medtronics, the Strykers, some of the larger organizations. They want to sell new devices to these hospitals and to the surgeons, to the clinicians. So by reprocessing, the cost to the surgeons and the clinicians and the hospitals is reduced, which in turn reduces the cost for patients that have undergone a procedure.
Jonathan, is the original manufacturer doing the reprocessing, or is it a different company?
So the original manufacturer, it's interesting because for a while it seemed like they weren't as interested in becoming involved in the reprocessing of their own devices. And what had happened is a lot of their own devices were taken into reprocessing facilities who obtained the appropriate clearance and started reprocessing and selling those devices to the hospitals. And so I think they're now interested in that because they understand that their business is being cannibalized by reprocessed devices, which are more affordable to the hospitals. So it's just an ideal scenario for the reprocessors, not so much for the individuals who are only focused on manufacturing new devices. That's a great question.
So are these devices used only once on one patient, or Does the hospital recycle them?
That's a great question as well. So the devices, depending on, so I mentioned that design feature, right? So when they're going through the design plan and what they intend for these reprocessed devices to be used for, they determine the amount of cycles that they could be cleaned and reprocessed. Some might be reprocessable a single time. Others may be reprocessable up to 5 times. And I think a great example of something that you could reprocess 5 times would be a cable. So I've got an example of a cable. This would connect to a EP catheter. And so this cable here, it's used in a surgical and in a clinical setting, and then it's typically disposed of in a biohazardous container. And so the problem with disposing any device or any material into those containers is it becomes contaminated with microbiological materials as well as chemical materials. Now, when you look at reprocessing, these cables can become contaminated with chemical contaminants because you might be using a specialized wipe to wipe off that soil that had accumulated over time or at the hospital before it was delivered to the reprocessor. So depending on what the instructions for use are for the individual device, you have the ability to reprocess and recycle several times or only one time, and that's dependent on material compatibility. So let's say you autoclave sterilize something 5 times, you might be degrading the material after cycle 5. And there's certain tests and procedures that us as testing labs are responsible for that are called cleaning validations. And those validations, and they're reprocessing validations as well, but the cleaning validations basically determine the amount of times that these devices can be reprocessed and then cycled back into the clinical setting for use.
You mentioned 5 times, several times. Is that a magic number, upper limit?
So I think 5 times is I think it depends on the medical device organization. So if you're a cable company and you make EP cables for the electrophys catheters, I think it's an ideal thing that you'd be able to reprocess that several times. But now if you're let's say you're working with catheters, which are more prone to issues after several cycles of reprocessors, then maybe not so much. So. And I kind of touch base on the commonly reprocessed devices. I mentioned endoscopes were one of those items. And I also kind of divided this table up into your complex reprocessing scenarios, as well as those that could be routinely processed in-house. And so surgical scissors, right? Any scopes, drill bits typically can be reprocessed in-house. But then, we mentioned the catheters and the other things that have complexities that they have to go to another facility that specializes in those techniques. And so here's just to show you some of the complexity of these catheters. And we've worked with, I think, every single one of these catheters before in a testing setting, not in a reprocessing setting, but they have features that are at the distal end of the catheter, which are prone to being damaged when they're collected by a hospital, because you have to train the individual who's used the device, typically it's not the surgeon, to dispose or discard the device appropriately. So, the device has to go into a certain packaging, or it has to be quarantined into a certain area, and if those steps aren't followed, then at the reprocessing facility, you find a lot of these devices that are returned from the clinical sites or hospitals are rejected and they're no longer used. And so a lot of reprocessors have a rejection steps in place that say, okay, if there's a bend or a kink on this particular portion of the catheter, then it needs to be rejected and not brought into the reprocessing process, right? Or cleaning, whatever it is that they might do. And cleaning, it could be as simple as you're flushing lumens of the catheter out with water, or you're implementing a enzymatic detergent and ultrasonic bath to essentially debride the contaminant material from the device. And it goes through several rinses and phases of that until you have a device that has acceptable levels of cleanliness and is capable of being reused. And I talked about sustainability as well, because I think that is kind of a key, it's a lot of the key point, it's a key selling point for the reprocessors, right? So it's a sustainability practice, but on top of that, I mean, you know, we see a lot of waste and medical waste that's cropping up, and I know that's, it may not be as much of an issue in the United States, but it is issue, you know, it can be a problem in other countries that may not have, you know, regulated waste procedures in place. So, you know, we have somebody that comes to the lab, picks up the boxes of all of our medical waste, and they dispose of it appropriately. You know, there's a logged record and a chain of custody, and that's all very well governed and regulated. But in you know, other countries, it may not be the same way. And so, you know, cutting back on waste is one key part of this. So you're not just throwing away, you know, a bunch of trash and you know, packaging and things of that nature. So that's kind of key. And obviously landfills and incinerators. Incinerators are where a lot of our medical waste or other biohazardous waste ends up, right? It's incinerated. So supply is strain as well. I talked about that. That's if this device, for whatever reason, has a rare component that's not readily accessible, you want to find a way to reprocess that device so that it can be brought back into the clinical setting, so that we're not waiting. You know, it's like a transplant patient, you know, waiting for an organ. You don't want to be waiting for a medical device. That shouldn't be the case. So again, this reprocessing is sustainable in that sense. And obviously, you know, if you're manufacturing from raw material, there's a lot of effort that goes into that. And that's a difficult thing when you're talking about energy or the water that's used, or if you're talking about the input materials. So that also cuts back on that. And you know, the key point here is safety. Both reprocessors and manufacturers are governed by the same exact guidelines, and the devices have to go through the same approval and regulatory processes. So you can ensure that devices that have been reprocessed or devices that have been newly manufactured are going to be safe for use in a clinical setting. And in some cases, we've seen that reprocessed devices are actually have a higher level of cleanliness than newly manufactured devices. Or they'll have different problems associated with those devices that weren't associated with the newly manufactured device, or you know, vice versa, the reprocessed device. So how does this substantial growth impact the industries? And I mentioned sterilization and testing laboratories were the key things that were impacted. And so sterilization is a big piece of, I'd say it's a key component of medical device manufacturing. Because if you can't get into a sterilizer and you can't sterilize, you know, several pallets of your device, maybe it's a bandage or a dressing, those go to sterilizers as well, you're going to not be able to release as much product as you expected. You're going to have delays in your lead time to your distributors, to the clinicians themselves, or healthcare systems. That becomes an issue. And so, you know, some of the challenges that face the manufacturers are the lead times, because if you're dealing with, you know, different types of sterilization that's required to sterilize the device, maybe because of material compatibility, you can't use steam sterilization. You have to use ETO. Well, ETO sterilizers right now are heavily bogged down with device and samples from some of the largest organizations. Largest organizations get into the chamber first, typically, followed by the rest of the, I would say, smaller medical device companies. And so individuals who come up with, maybe it's a compelling or a unique innovation that might improve patient outlook, sometimes is delayed to market because of, you know, this fact here, sterilization. And one other interesting thing to note about sterilization is, you know, you might think that the device itself is placed into a sterilization chamber, right? But the entire device and the packaging is palletized, and inside a carton or cardboard, it's placed into these chambers and the sterilization occurs. And so there's a few key things that I would say are relevant to testing because of this feature, and a lot of the time it's you know, residuals from those sterilants that are being used that might be on the medical devices. So we have to monitor that as a testing laboratory, and it's not only our responsibility, but it's also the responsibility of the device manufacturers and reprocessors.
What is the sterilization process? Is it steam only, or is it
Yeah.
other materials?
Yeah, I'll jump right into that. So we have, so just to talk about, you know, the types of sterilization, right? You've got ionization, radiation. hydrogen peroxide, so they vaporize hydrogen peroxide. This is kind of I would say, a new method that's being utilized that has some material compatibility issues with certain devices. So you're getting interactions with the device material or the polymeric material and hydrogen peroxide. That could be an issue. Ethylene oxide is the primary, I think I have right here, right? 50% of all disposable medical devices are placed into a chamber and are exposed to ethylene oxide for a certain amount of time. Then it's degassed again for a period of time, and those devices are sterile until tested and documentation indicates so. And so something that's interesting about ionization radiation is, you've got your gamma, your electron beam. That's I would think of it less of a chamber, but more of like a conveyor belt type of thing. So you have a conveyor belt that is moving now boxes. Instead of pallets of boxes, it's moving items through that ionization or that field, right? So that you can achieve the sterilization of that device through packaging. So think of it of like an airport X-ray machine, right? So you're putting your luggage in through there that's getting exposed to similar beams that these types of medical devices would be when they're undergoing ionization radiation. And a big thing here too to mention, so although this is the largest method of sterilization for medical device companies, there's a lot of stink that's been raised recently about ethylene oxide, and they're concerned about the ethylene oxide exposure. So let's say you live right down the road from an ethylene oxide facility, and they don't capture all of the ethylene oxide. I mean, they're making technologies now that can do that and can recycle it, but that could be getting into the atmosphere. And there's been some concerns that have been raised by certain bodies and health organizations that are concerned about individuals' exposure to ethylene oxide for those living near these facilities. So I think Florida is a great example of this exact phenomenon. There's a lot of work that's going on in Florida, and there's a lot of ethylene oxide sterilizers in Florida. So I think that's why it's kind of stemming in that particular region. Perfect. So, and we talked about the different types of sterilization, and I want to talk about what they might be ideal for. So we talked about material compatibility, right? If you have a polymer or something that is not very heat resistant, you might put it in the autoclave and you open the door and it's melted, and that's an issue. So they kind of have to determine what the best route of sterilization would be. And so I'd say leaving behind very little contaminants on the device itself. I think radiation is a very promising form of sterilization. It's also very effective. And again, hydrogen peroxide and ethylene oxide can leave behind residues, although it's, I would say, very well commercialized at this point, and they have several facilities that can, for the most part, keep up with the demand coming from med tech companies and reprocessors. And so just some considerations as well. So these take a long time compared to steam autoclave. Your autoclave cycle might be 30 minutes, but then you're kind of constrained on how much you could put into the chamber. Same thing goes for ethylene oxide, except their chambers are the size of airplane hangars. And they do make autoclaves that are the size of airplane hangars as well, but you're not going to find that in a hospital setting, right? Ethylene oxide is really your key contributor to sterilizing these devices. And I've got some images as well just to give you an idea. So this is very much enlarged, and it is nothing to scale of any of these processes, but this would be an example of an autoclave sterilizer you might see when you're going to get your blood drawn. You might see at the dentist, right? These sterilizers are going to be present. They're used for primarily quick sterilization of the instrumentation. Although we use them in the lab to sterilize microbiological media that we might use for growth promotion studies or any of the other microbiological tests that we perform in the lab. And then ETO, so it's it's very complex. There's a preconditioning phase. So you heat the items up to a certain temperature. You place them into the chamber, and then ethylene oxide gas in a certain concentration is flown into the chamber, and then that is they use a vacuum pump to basically pump that out, and it sits again for another hour so that it can basically leave the packaging. Because the idea of heating these things up, especially in these pouches, is that you need whatever method of sterilization that you have to penetrate the pouch material. And so something interesting, most pouches are Tyvek, right? So there's a certain layer of Tyvek, but these types of sterilization methods, ETO and hydrogen peroxide vaporization, can penetrate into the packaging, access the device, and then through some reconditioning phases, leave the packaging and the device, leaving behind very minimal residue. This is so your gamma sterilization, right? I just kind of imagine the conveyor belt. So, an Amazon warehouse, if you will, of boxes all going through, a gamma beam, as well as electron beams. And so there's a source that generates that and obviously provides sterilization of those objects. Hydrogen peroxide, I would say, works very similar to ethylene oxide, except you're using hydrogen peroxide instead. One interesting thing, so I brought a golf ball with me, and the reason I brought this with me is it's been E-beam sterilized. So, and this is the ball I bring out on my golf outings because I can hit this thing further by about 10%. All right, and so I'll pass this around later, but it does. So they E-beam sterilized these golf balls, and this was something that was done in the 60s by Oak Ridge. Actually, they had the furthest drivable golf ball that they could make, and they used a different form of irradiation to basically compact the core 95% and make this thing fly a little bit farther. So when you deal with E-beam sterilizers, which we deal a lot with E-beam sterilizers that are involved in the cleaning validation process, you end up with a lot of E-beam golf balls. So just something interesting though. I had no idea before I was dealing with that industry, and that was something that they do, but that's what they give out at their shows. They're at the conference and they're like, hey, have a golf ball, and I'm like, well, what's the deal with this? And it's like, oh, you can hit it further. I'm like, that's all I need to know.
Can you prove that it went through that process?
That I cannot. It says so on the golf ball, so I'm just taking them for their word. I don't know.
Did it help your game?
What's that?
Did it help your game?
My game is still terrible, so it didn't help me at all. So kind of shifting, I know I spent a lot of time on sterilization, but that's an important piece of medical device manufacturing. But I mean, testing really falls right under that, into that next slot there. And as you notice, it's something that's done pre-sterilization and post-sterilization. And you might ask, well, why are you going to do this pre-sterilization if you're just going to sterilize the device and then examine for contaminants after it's sterilized? And the reason is because when you validate your sterilization cycles, there is a certain microbial load that's present on the devices that the sterilization technique or the parameters that are set is effective at basically full killing, right? And so if you have a bioburden, right, so a microbiological contaminant that is in excess of the coliforming units that were on the device when you validated it for sterilization, you could find that even after sterilization, that some of these contaminants can reside on the device and can be recovered using some of our testing procedures we utilize in lab. And again, the demand is crazy because there's been, as devices go in pallets, they come off pallets, go back to the facilities, and they're sent immediately to these testing laboratories. And in the space, there's a few large testing laboratories, but there's a lots of smaller laboratories cropping up that are offering better turnaround times, which is really the key, I would say one of the key selling points, because you used to be able to get a service done from these large organizations in 3-5 days, but that's extended out to several weeks. And so it's just become unreliable to deal with a test partner that is not capable of producing the turnaround times that they once were able to. And turnaround times are very much dependent on the methodologies that are used, because there's obviously constraints. For example, if you have to incubate a plate to examine the count, right? A total aerobic count. That's going to take 3 days at a minimum. That's what USP indicates. That's what some of the other regulatory bodies indicate. So we are constrained in turnaround time, but it doesn't mean that we have to release these results several weeks later. The work can be done if the test articles are in the laboratory. And that goes hand in hand with timelines. If you're waiting on testing results to come in and you're waiting on your laboratory to present that information or that data to you, you don't know if you can release these products to a distributor or a hospital that's expecting them to arrive today. So that definitely becomes an issue. And in addition to that, so it's not only manufacturers and reprocessors, but it's also healthcare facilities. So
How close are you to being able to prove that it is disinfected electronically rather than going through a 2 or 3 day petri dish kind of thing?
yeah, so it depends on what the contaminant is. Biological contaminants, so sterility testing, just to give you a sense, they have a sterility testing or an assurance level of like 10 to the -6, and that is deemed acceptable for a device that has been sterilized by one method or another. So if you go through this process, you have to use the accepted procedures that are been demonstrated in standardized methods. So a lot of the time we have to follow the methods that take 14 days to evaluate sterility, particularly for microorganisms on a device. So we are constrained by that. But now if you're talking about, they are getting close to what was be called like rapid sterility testing. So as opposed to 14 days, you might have your results in hours, but you have to look and you have to put yourself in the shoes of an FDA auditor because they are familiar with the method that takes 14 days, not familiar with the rapid method. And so you're kind of trying to, have a paradigm shift from, hey, this testing takes a long time. We have a rapid method and, we're seeing similar or acceptable results by performing this method, but it's not necessarily accepted 100% by FDA or other governing bodies. And same thing I would say goes for endotoxin. So endotoxin is interesting because you're using horseshoe crab blood essentially, and so it's an animal-based material to perform these assays. And the thing that's interesting about that is, hey, we have special recombinant or otherwise synthetic forms of that reagent that could perform the same assay, but nobody wants to switch over to that because they're concerned about how it'll be received by regulatory bodies when they're submitted.
Well, that's one of the problems with ISO, is it's so rigid that it's very difficult to improve it.
Yeah, absolutely. So, I mean, I think there's geniuses and great minds out there and researchers that have come up with very interesting methodologies, but they can't, fully be deployed in a highly regulated industry, especially like medical devices. So you might be able to get away with a unique or a fast method for environmental, although environmental is governed by some certain standards. Certain things, especially depending on the criticality or the classification of the medical device, they typically don't allow that. So, and then I've got, so the categories of required testing, right? We talked about bioburden, which is microbiological based. Something I don't talk about much in this presentation is biocompatibility. That's more on the animal testing side. We're very much involved in laboratory testing that does not involve animals. And so these tests still need to be performed by medical device companies, but we might work with somebody who provides biocompatibility services to supply those services for our clients, as opposed to directly getting involved in those processes, having a vivarium, all of that good stuff. And endotoxin, sterility, so talk about sterility, again, I think those are the two key methods where, hey, we have a different alternative. They're just not currently being deployed in the industry. And particulate matter is another large thing that they're looking for in medical devices, because if you're generating it from a raw material, or if you have some kind of extrusion process you're using to manufacture the device or make the shaft of the catheter, Particulate matter could be a huge issue, and above certain micron size, I think between 25 and 100 microns, that's when you start talking about you're getting into embolism territory, and so you don't want to introduce a device into the bloodstream that has that type of particulate matter, affixed to the outside of the device that could be shed into the bloodstream and then cause those types of problems for patients. And I mentioned the cleaning validation and reprocessing validation. So that's where we look at how many cycles the device can be reprocessed, and then we pick maybe 3 tests or 3 different cleanliness evaluation parameters, such as bioburden, endotoxin, or TOC, just to make sure that after the reprocessing or after the cleaning, those types of contaminants are not present. And we'll do that through each cycle. So we're evaluating, at cycle 1, maybe midway through at cycle 3, and then after cycle 5, we're able to determine if those are absent, or if you're using like a cleaning solution, if they accumulate over time as you reprocess the device. And why testing matters is, less recalls, less infections, or loss of market access to the product. So kind of similar to sterility, this last part, but the recalls and the infections portion is really what testing of these devices prevents. And it's part of the reason why FDA regulates it, and it's part of the reason why for certain classifications of device that these companies take this seriously, and they definitely do document and prove to the auditors that these devices are clean and effectively can be deployed again. And so it's a key step, right? I've mentioned this, and it goes through, there's kind of several phases here that you can follow, but regulatory approval being the last one. And obviously to obtain this, you have to prove that the device is truly clean and is functioning if you reprocess it as a newly manufactured device. And testing labs were very much, just like sterilizers, we're embedded in the life cycle of these medical devices, right? They're sending them to the sterilizer is sending them to us, the medical device manufacturers are sending them to us before they go to the sterilizer. So we're giving them information on their process, we're giving them information on the effectiveness of their reprocessing or their cleaning. So there's several things or pieces of information that can be gleaned from that. And we work and we collaborate directly with engineers, quality managers, and regulatory affairs. So you might be in a meeting, you might be explaining results to an organization, and a lot of the time, all of these individuals at that organization, they're going to be present on the call, and they all have questions that are related to whatever role it is that they've been placed in there. And I think I find the enjoyment of working with engineers because they're always working on some cool process, or they're coming up with some unique techniques, and that's, I think, more of what Bioscension can help with as well is, hey, our cleaning process isn't working right now, or our manufacturing process, it's putting too much heavy metals on the device. I think through testing and then obviously collaborating back and forth with them, we can help them improve that process. And obviously those companies really love that type of interaction. And I think that's something that's less so now that testing labs have become so busy with all the samples that come in. They just report results and they leave it at that. They don't essentially try to help these clients improve their process and have a successful product. And speaking about contaminants, right? So going back to contaminants, it's like how do they accumulate them over their lifetime, right? And throughout their life cycle. And here's a huge laundry list of contaminants that the FDA either regulates or AAMI especially has procedures around for testing them. And I'm not going to go through every single item on the list, but just so that you've got the biological side of things as well as the chemical side of things. And chemical being if you're using detergents, cleaning agents, that type of thing, that's going to end up potentially on the medical device. Microbial contaminants, that's typically coming from extruders that have biofilm buildup, or they've been returned from a hospital and they were sitting in a biohazard container for a couple weeks and they might have been in the Arizona sun or something like that. So those types of things could lead to the microbial contamination. Particulate matter, I think of cinnamon. You know, those applesauce pouches recently. They had an issue with the cinnamon that was placed in it having too high heavy metals, and that's from the burr mill that they were using to grind up the cinnamon and then put into the product. So, you know, they wouldn't have been able to identify that without testing, but it's very much similar to what we deal with in medical devices, except it's more or less plastic fragments or something like that you might experience. Metal shavings could come from the machines themselves, obviously, as well. Human origin contaminants, that's, again, very microbiological based, and this gives you an idea of, you know, where those human origin contaminants can come from. And this image here is a very poor example of how you should dispose of your sharps, so just keep that in mind. Sharps should be disposed of in a sharps container and not in the biohazard container where, you know, you might be stamping down the material with gloves or something like that. But I mean, it gives you an idea of what you might see at a healthcare facility where, okay, hey, we finished using this device for surgery, we're going to dispose of this in the biohazard container, and we're going to send it off to the reprocessors, you know. So reprocessors get a bag of blood and device, basically, and they've got to, you know, pick through that, reject certain devices, and determine which ones can or cannot be reprocessed. So very, very dirty job on the front end for the medical device reprocessors. And this is just a great example of, you know, either a manufacturing or reprocessing facility. You see there's a lot of machinery that's involved in that, and that would obviously lead to the particulate matter based contaminants, as well as some of the chemical based contaminants. Let's say you cleaned, you know, this machine with bleach or something like that, and then you ran a batch of devices through the instrument, and now you've got an issue, you've got a chemical contamination problem because of the cleaning technique you used. And so again, just I think this helps kind of visualize, you know, the types of items or the contaminants that you can find on a medical device, and it's easy to see, you know, based on this image, how those types of things could find their way and adhere to the surfaces of those medical devices. And I mentioned healthcare facilities as well, but healthcare facilities, there's a couple nuances, right? So the biggest problem with healthcare facilities would be environmental and would be water-based, you know, issues that they encounter. And so the water-based problems would be used in their endoscope reprocessors, for example. And if you don't have a purification system that's basically up to spec or is routinely monitored and managed, then you can introduce some of these chemical residues as well as some of the waterborne contaminants. And Legionella is a great example of what hospitals are constantly dealing with and are monitoring using, you know, immunological assays and other forms of assays to make sure it's not in the water. And, you know, just to give you an idea of this is an environmental setting, right? So we clean the bed with a certain disinfectant, and for whatever reason, it doesn't perform a kill on that organism. And so that stays behind, and, you know, that's where we see the rise of antibiotic resistance or, you know, organism resistance to a certain cleaning agent. And these hardy bugs can definitely cause infection and disease in individuals that are treated in these types of settings. So it's really important that, you know, there's certain steps or procedures in place at the hospital to make sure that these things are being done appropriately. One thing that you may not think about as being a key issue, but the HVAC system. So if you've got a condenser coil and you've got this in your healthcare facility and, you know, it's a 100,000 square foot facility and it's moving a ton of air, you can have some serious issues arise from your HVAC system, and that's going to be putting, you know, microbial contaminants, particulate contaminants into the air, and that's what we can capture using environmental monitoring. So we do a combination of both water monitoring and environmental monitoring in these hospital settings, just to look for the types of microbes or the disease-causing elements that you might encounter as a patient obtaining some kind of procedure or just going to the hospital, right? And this gives you an idea of, we're talking about water purification systems. It's like, oh, well, it's easy. It's just a reverse osmosis. You know, I've got a couple of filters under my sink. I can turn on the tap. I get pure water. But, I mean, it's a lot more complex than that. They go through a lot of polishing stages, right? There's activated carbon filters. I mean, this water has to feed to sterile processing departments of the hospitals, as well as some of the taps that are accessible in some of the operating rooms or, you know, some of the rooms in general. So, you know, there's a key infrastructure And it's very difficult to keep track of and monitor, but it is possible with the help of the maintenance facility and the directors. But these types of things need to be monitored as well. And so we're doing pre-filtration monitoring, we're doing post-filtration monitoring as well. And we're looking at, the point of use. What does the microbial content look like? What does the endotoxin look like at the point of use? We want to make sure that that's low and it's within acceptable levels. And so I mentioned the resistant Staphylococcus aureus, ionic contaminants which can be found in water, and heavy metals is a great example of something you might find. Your chloride, nitrates, phosphates, those are some items that you might find that could be in elevated levels that could down the road lead to issues like, for example, scaling in your water system or your water purification system can be a problem. And if you can identify what ionic contaminants are high or at elevated levels, you can prevent those types of things happening to your system and keep it in peak operating condition. And Legionella, of course, the last one. That's just any hospital you go to, they're looking for that. That's for sure. And so, what do we test? And this list is not I would say it's not a full list at all, because to list out all the test services and all the particular analytes or things that we're looking for, it might take several of these slides. But I just wanted to touch base on some of the key things that we perform. And I would say these tests are they're heavily utilized for cleaning validations and reprocessing validations. And so, you have a protein contaminant, endotoxin contaminant, or any other contaminant, and you have a particular test method, which again is governed by those standards or standard techniques and methods that we follow to evaluate that. And I wanted to talk about what the impact would be if that's uncontrolled, right? So, endotoxins, pyrogenic reaction, that can be an issue that you encounter if you have high levels of endotoxin and it's being introduced in the body. And I would say even in small quantities, we're talking about nanograms to micrograms, can even cause these types of reactions in patients. So small amounts of this, especially in cerebral spinal fluid contacting devices, it would be a very bad thing.
It looks like some of those tests would take more than a day.
So here's the great thing about, so bioburden, yes, I agree with that. That's about a 5-day to 7-day process, because bioburden looks at a couple of things. It looks at your aerobic content, so your aerobic contaminants or aerobic bacteria, and your fungal or your mold, contaminants. And so fungi plate counts take about 5 days to incubate, and that's relatively at a lower temperature compared to your membrane filtration or your growth promotion on tryptic soy agar for aerobes, which takes maybe 48 hours to 3 days. So these definitely take some time. But I want to, and when I move forward, I've got a couple of interesting instrumentation and assays and some kits that we can use to turn this type of testing into, maybe a 3 or 4 hour process, and I have the results for you. And then we're talking about, protein contamination. Hemoglobin is another key analyte that we look for, and TOC. And so with the, you think of TOC, hey, that might take a long time. I've got to perform wet oxidation on the bench, or I got to, perform some kind of titrometric chemistry. That's actually not the case. We, especially with the invention of these analyzers. So they've got special analyzers which we can get down to insanely low detection levels. We're talking about parts per billion, parts per million, right? You're injecting a sample directly into the machine. Everything's automated, and it spits the results out. And then we perform calculations on our end and generate the reports. So definitely some of these tests can take a culmination of days. But some of these tests, if we're using micro test plates, which for protein, endotoxins, hemoglobin, we use micro test plates and micro test kits. We use a spectrophotometer, and it basically it can scan 96 wells. We implement a standard curve into that plate, and then we're looking at all of the samples that we plated on that as well. And so we can get a really good idea of what that level of contamination is in the form of concentration, right? So we're looking at micrograms of protein per milliliter. And then for device, what's more pertinent is the surface area of the device. So micrograms per centimeters squared is what the regulatory bodies are interested in seeing. And so there's some calculations that go on that basically can get you to that form or that unit. And I've got an example of that in some coming up slides. So what is it that we use? Kind of, similar to the question that you had asked here, there's some different methods, and the ones I want to key in on are these assays that are performed using spectrophotometer, and particularly the microtest reader, the microtest plate reader. And so, you have assay preparation. The plate basically is inserted into a tray, and then that plate is then scanned, and the results are presented on the terminal or the PC for you to basically evaluate and then analyze. And so that makes it very easy to come up with the results in, I would say, less than a day's time for these types of contaminants, right? Your endotoxins, protein, hemoglobin, TOC included in that as well. TOC though is analyzer based, so you kind of just plug and play, and you're seeing results after it runs the analysis. Particulate matter, I would say that's a relatively quick test as well, because you're using a very similar technique that you'd use for bioburden, and it's called membrane filtration. So you're basically taking liquid, you're filtering it through a filter paper that has a certain porosity, and that captures certain particles, and it lets fluid go through. And so the particles that we're interested in are the ones that it captures. And then we can use either so you can use microscopic techniques, which would be the membrane filtration based technique, or you can use light obscuration techniques as well. And that can give you an idea or an insight into the particulate matter that's present in a device extract. Because I'll talk about the complications of getting these things off of the device so that you can analyze them effectively. And then you need to understand, is it the extraction method that we use, was that efficient or was it not? And so we have ways to evaluate that also. And so the microtest plate reader, I think this is a great image of what you could utilize. And this is basically it's a benchtop spectrophotometer. And so I'm sure you may be familiar with the cuvette, right? A 1 centimeter cuvette. You can put that into the cuvette holder. It passes a light source through that. There's a reference light source. It captures the information in the form of absorbance. And you can basically ascertain measurements from that utilizing calibration curves and standard curves that are developed. And this is a great example of, so there's the cuvette. Here's your microtest plate. So the microtest plate has 96 wells. Some can have as many as 384. But I only prefer to pipette 96 times, so we go 96. 394 is going to be more a robotic technique, so that's something that you'd utilize a robot. There's automation that's involved, and you can perform an insane amount of tests, high throughput, utilizing a 384 well test plate. However, 96 is great, and we can do anywhere between, let's call it 25 to 30 samples on a single microtest plate. And so these are the kits that are involved, right? I mentioned the endotoxin, the KCA assay, right? So this assay takes about an hour to run after you've set it up. And you have control standard endotoxin here, which is basically used to make your standard curve. And then you have the LAL reagent, which is isolated from the horseshoe crab, that you basically inoculate and you react in each well to perform this assay. And the assay is kinetic, so it's taking 121 measurements, and it scans every single well over the period of an hour, and it basically generates a curve that you can utilize and reference back to your calibration curve to determine the concentration. Protein assay works in, I would say, very similar manner, very similar format. Same goes for hemoglobin assay. The only thing that's different between these kits is the preparation instructions and the reagents that are used to basically carry out these reactions or stop the reactions so that you can read the microtest plate. Here's this is a great example of the TOC analyzer that we have in our laboratory. So we've got the auto sampler here. This is going to be where the wet oxidation and the chemistry is performed, and then the analysis is performed as well. And off to the right would be a PC, and that's where you're getting data collection, and you can perform the calculations on what the parts per million of total organic carbon, so when I say TOC, total organic carbon, is present in a water sample or a medical device extract. this was the, basically it's a method of membrane filtration. You can use it for bioburden, you can use it for particulate matter, but it gives you a really good visual representation of, how this takes place. So, you're putting you've got basically a filter apparatus, the membrane filter gets, kind of smashed or pancaked between that apparatus, and then a vacuum is applied, liquid is added, and then the contaminants are collected on the surface of that membrane filter. And instrument detection, right? So we're looking at parts per million, parts per billion, parts per trillion. And I just want to put this in perspective, right? So how small is a part per million or a part per billion? And what's the detection capabilities of these instruments? And that's actually, I think, well represented by this example, and this was kind of how it was taught to me in university as well. But in a single Olympic swimming pool, so 600,000 gallons, 1 part per million would be 1 1/4 2-liter bottles of water. And so that instrument is essentially sniffing out that analyte. If it's well homogenized Olympic swimming pool, it's sniffing out that analyte at that level. Now, parts per billion, 1/2 teaspoon into an Olympic swimming pool, the instrument is capable of detecting. And so the instruments that are most capable of detecting this would be the TOC analyzer. Parts per million capability is in the spectrophotometric techniques that we utilize for sure. But then parts per trillion, so we're talking about nanograms now. This is a 1/2 teaspoon into 20 Olympic swimming pools is the detection capability at that level. So it's impressive how far the instrumentation, the detection capabilities have come. And just having something that you can set up on your benchtop, input a sample, and then obtain this type of information. Is it there or is it not there on such a small quantity? I think is a very powerful tool. And so existing test methods, right? So this is where, okay, you've got a medical device and I need to perform membrane filtration. You can't take the catheter and just run it through the filter, right? There's no way to do that. So you've got to come up with a means of doing that, and that's through extraction. Typically we'll use water, so like ultra pure water to accomplish these extractions. For bioburden, we use what's called a recovery eluent or diluent. And so that basically revives any microorganisms that are present and also extracts them from the device. So we're not directly testing the device in most cases. We're testing extracts of the device, and there's procedures that are in place to basically accomplish that. So if you've got lumens, you can flush the lumen with that extraction diluent or water, for example, and collect it into a sample container. If you've got, let's say something like this device here. This has lumens, so you would flush this, but you would also perform immersion and sonication. And so to do that, you need to find an appropriate size sample container. The sample container needs to be sterile, needs to be pyrogen-free, so it's difficult to find those types of, you're constrained very much by what sample container you use for these test procedures. And kind of going into the test methodology, so a lot of these methods were specialized or adapted from existing microbiological method or chemical analytical methods, and a lot of research has been done to accomplish that. And so one key thing you might run into as well is interfering factors. So you extract everything from a device, you might extract the coding from the device and then perform a test and then find out that this is inhibiting the endotoxin response completely. So you need to figure out ways to basically get around that or cut out the inhibition factor without manipulating the sample. So that's kind of the key thing. You can't, no mitigation strategy used can manipulate the sample or change it in any way. So you need to come up with ways that basically mask certain contaminants or certain interfering agents from the assay that's being used or the chemistries that are involved in that particular assay. And so here's a great example, because I've got this container. By the way, this is 120 milliliters, so it's a little bit smaller than that coffee cup. And we've got a device here with a handle that's maybe a foot long, and we've got tubing that, who knows, it might stretch all the way across this room to that wall. And so the thing is, well, how do we extract this device into this pyrogen-free container for LAL testing? And I mean, the way to do that is through a unique test setup and kind of having some rigs in place. And fortunately for this particular device, we can inject fluid through the tip all the way into the lumen, and right hiding under here is an IV spike, into the IV spike as well. And so we perform static incubation on the device while the extraction fluid is contacting the lumen pathway of the device. And then after that extraction procedure, it's about an hour, incubating at 37 degrees C. So again, biologically relevant temperature. It's basically ejected or flushed into this container. And we take that extract, and that extract is actually what's tested. So you are challenged all the time with different devices and configurations of devices to figure out, how do I get it into this container, or what container can I even use to perform this testing? And measuring device cleanliness, I kind of touched base on this already, but we talked about the extraction methods. So there's different extraction methods that you can utilize. We have a lot of different tools at our disposal. Ultrasonic bath, oscillator shaker are the go-tos. Those are the workhorses of the laboratory. And then static incubation, right? You can place it in an incubator. They make shaking incubators, so you could shake and incubate at the same time. So there's different ways to extract those contaminants from the medical device into a sample container, and ultimately the sample extract that you're going to analyze. And here's the surface. I mentioned surface area matters. This is kind of the key point of the slide. So if you've got a device and it has a particular surface area, that's provided by the manufacturer, thankfully, because there's a lot of complex regions on that device. That information goes into the total calculation of, that's going to be basically supported or documented for regulatory support, right? So the device is 240 centimeters squared. You're extracting 100 mils sterile water. Here's the result that you get on the analyzer, but then you have to calculate that, and you have to take into account the surface area. And so this buys, I would say, medical device manufacturers and reprocessors a little bit of wiggle room, because the larger the device that you have, the lower that you can get this number here. And obviously the lower the better, because acceptance criteria is already very stringent and very tight on these items. And I've got just a quick overview of what's happening when we take the device extract. So the device has been extracted, it's in the container. We're taking aliquots of that. So if we're microtest plating it, we're using micropipetters, putting it into a microtest plate, where we can pipette it into a TOC vial. We can place it into a membrane filter. We can perform all the tests that we're interested in performing, from biological to chemical, you name it. Then we generate some raw data after testing. That data is analyzed, and then the results are reported. Those go on to the client or the manufacturer. And this is that's the key piece. A lot of the times, it's
Yeah, I'm just curious. You talk about how prescriptive, the regulatory bodies are, but it sounds like you have a lot of free, decision making on how you collect some of the, specimens. So I'm surprised they aren't specifying that.
Yeah, so they somewhat specify that in the sense that they'll say, okay, because I mentioned 100 milliliters of water was used to extract in that example, but really it's 3 milliliters per centimeter squared of device, or 6 milliliters per centimeter squared of device. So that gives you an idea on your extraction volume that you need to utilize to basically perform the extraction. And then there's additional standards. That's kind of where USP steps in. USP comes in and they say, well, we want the containers to be non-pyrogenic, and it needs to be a certain material, right? Because polypropylene, for whatever reason, might adhere endotoxin to the walls of that container. So when you're testing it, you're not actually getting a representative value of the amount of endotoxin present in that sample. So there's definitely, I would say, there is some free or wiggle room and some decision making in that, but there is still some very descriptive standards that indicate, hey, this is how it's been tested, here's how we would like to see it tested. And then when they're doing 510K submissions or any other submission, typically it's going to have to follow those guidelines. Otherwise, the testing can't be deemed as, hey, this was USP compliant with bacterial endotoxin USP 85, or this was compliant with the bacterial endotoxin USP 161, which will tell you for cerebral spinal fluid contacting devices, there are certain testing constraints that we have to follow. So. And so how clean is clean enough? I've talked about cleanliness this entire seminar, this presentation, and I don't think I've provided an idea of how clean is how clean enough. And this is the typical acceptance limit, and these are for the analytes that are going to be utilized in cleaning validation studies. So we've got your protein, hemoglobin, TOC, and the very interesting thing about these is we can simulate protein, hemoglobin, and TOC on devices without them going to a clinical setting. So we can create an artificial test soil, we can inoculate the device with that test soil, we can then extract the device, or we can clean the device, and we can see what levels of analytes that we're looking at based on that artificial test soil composition. And the typical acceptance limit, right? So this is again in per centimeter squared. It gives you an idea of how many micrograms per centimeter square are allowable on these devices. So it's not zero. There is a number, there's a defined number, but what they suggest is at or below this number that the devices are not going to pose a risk to the patients for which those devices will be used on. So, and there's risk factors and other analyses that go into this, and again, this is highly discussed in those regulatory bodies and international standard organizations on what these should be. And these also, based on device classification, can change. So be aware of that. If it's cerebrospinal fluid contacting, it's a lot lower, sometimes 10 times less than you would see these levels for, let's say, an implantable or some other blood fluid contacting device. And here's actually, so a great standard to look at. So TIR 30 is now AAMI ST98, and that indicates for protein, hemoglobin, and TOC what the acceptance limit is and how you should perform the cleaning validations on those devices. So they do have some very, I would say, sound and robust guidelines in this particular standard, and that is, I would say, the golden standard for medical device cleaning validations and reprocessing validations. And I talk about the classification, again, this is really where it's, you'll see the 10x decrease from something being a non-critical device to something being cerebrospinal fluid contacting. And as you can see, the allowable amount of endotoxin is 2.15 EU per device for a CSF contacting device, but for your semi-critical, so these might be bloodstream contacting devices, it's 20. So you have a lot more room based on what you intend to use that device for and what it's going to be contacting on the patient. And your typical acceptance criteria, this is just a little guideline here, just indicates, okay, if you do microbial studies, CFU per device, that's what they want to look at, right? Endotoxin per device, or endotoxin per mil if you're working with a pharmaceutical suspension. Proteins, always microgram per centimeter squared. TOC falls in that same boat. Particles per milliliter is for your particulate matter, and they actually look at different size particles. In those standards, they might say, particles less than 25, there's X particles per mil. Particles less than 50, there's X particles per mil. So, and cleaning validation, again, a very key point in the of the process, and something that we perform frequently in the test laboratory. And we utilize an artificial test soil to inoculate the device. We take the manufacturer's instructions for use. So they'll say, use this detergent, soak it for this long, put it in an ultrasonic cleaner, and it should be clean, right? And dry it off and it's clean. So these are the types of steps that we'll perform in the laboratory. And then after that, we'll do the testing on protein, hemoglobin, TOC to evaluate that cleanliness. And all of these items are, kind of what I've touched base on regarding the extraction, the recovery of those contaminants from the devices. And so closing the loop, so testing isn't just compliance, it's optimization of the processes. So a lot of the times, companies will approach us and say, hey, we need this tested, and what they want to see is a report that shows that their limits of the contaminant are below the acceptance criteria per the standard. And a lot of the times that's great, but if you're working with engineers, which we do a lot, we work with those engineers, it's optimizing their process. So, and maybe somebody higher level at the organization is not thinking about that, but the process engineers are. and they're interested in the feedback and the observations the laboratory has. They're interested in the, the reports and the analyses, and they're very, I would say, invested in what it is that we're doing on the testing side of things. And just to summarize, so testing provides proof of cleanliness, your insight into process effectiveness, and then it also gives the manufacturer documentation of compliance. So when an auditor arrives, they knock on your door, you can show them the documents that suggest that, hey, these devices, they meet the acceptance criteria and the standards per ISO, AMI, FDA. And what it all adds up to, right? So you have your regulatory submissions, you have your evidence, and then your insight to prevent recalls and reprocessing failures. I think that's kind of the key step. The medical device companies don't want to get to the third bullet here, and that's why they perform the testing, and that's why FDA is heavily regulating this space. And again, just if you're looking at laboratories and what it is that we do for these companies, it's we measure, we interpret. We don't act so much, but what we do is we communicate that so that those individuals can act if there is an issue with the process. And it's more than a report, right? I mentioned that they just want to check off, they want to check box and say that this passes or it fails, but it's a lot more than that. And it's that communication, it's the insight into the process and the test methods that we tailor towards these devices based on their complexity that matters a lot, because these companies are responsible for developing a test plan, and a lot of the time we lend our hand to say, well, you should really evaluate this as well, because we suspect this will be an issue. We've seen this with similar devices of similar classification in the past. And at Bioscension, I think we do that really well, as we communicate, we collaborate. So we're not just a report the results, here's your report, here you go, have fun type of organization. So we really enjoy that collaboration. We enjoy working on new projects. And we're always designing new test and workflows that can be utilized for unique products that have all these complexities. Thank you.
Quite interesting and complicated. I mean, there's lots of issues.
Yes.
Any,
Yep, one question I have. This obviously operates in a clean room environment. What's the level of classification you operate in?
Yeah, so the testing itself, if you're performing sterility testing or microbiological testing, we operate in an ISO class 5. And fortunately for us, we don't have a dedicated clean room at this point, but we're adding a clean room, or basically a spot that we can perform sterility testing and get that sterility assurance level. But we use a laminar flow hood, and so it's basically keeping everything inside of the laminar flow hood at ISO class 5, but blowing everything else out into the laboratory. So, yeah.
so it's clean in that area, but very dirty elsewhere.
Yes. Yeah.
Mike, you got?
How big is your organization?
So we've got about 10 employees right now, about 5 analysts. We have, I would say, several large and small medical device clients. We have a few very large healthcare facilities that we're responsible for performing water monitoring on. So, and we're definitely growing. I'd say 2025 has been a growth year for us. So we're seeing a lot of growth and a lot of development in areas that we may not have expected to be our largest growth segments or sectors. Because when we set out, I mean, initially we started performing bioburden testing, and that's all that we did. And then we expanded from bioburden testing into cleaning validations and all of these other analytical tests or quantitative tests that we could state, what the level of that protein, hemoglobin or TOC is. So yeah, definitely growing, and we're adding several services to our scope this year, actually. The recertification audit that we have coming up, we're adding a laundry list of tests. So.
Okay. One question in the back.
How many projects or devices
Yeah.
do you have a failure that doesn't meet the specification limits?
Yeah.
Like
No.
a very small percentage,
Very.
25%?
Very small, but I will say it depends on the project. So if this is for R&D purposes only, you'd be surprised that the thing that the engineer thinks is going to work great doesn't end up working out as well in terms of cleaning the device. So we do see in R&D scenarios that some of these devices are failing bioburden primarily, and then in the cleaning validation segment, we're seeing TOC and protein being an issue. And for cerebrospinal fluid contacting devices, LAL is very tricky because there are several interfering agents that can interfere with that assay and can enhance the result that you're seeing, kind of presenting you with a false positive until you further mitigate that. And typically it's enzymatic detergent that's being used to clean those devices ends up interfering with those assays. So I would say it's a very small percentage when we're talking about lot release, and I think the percent that they target is like 1% or less. And then for the R&D process, that percentage goes up a little bit more because they're changing certain parameters about their cleaning procedure to basically develop something that, hey, this is how we're going to clean this cable or this catheter. So
Those are probably the best projects then, aren't they?
they are. We find a lot of recurring test samples coming in after that. Yes.
Okay, I'm going to insert one final question, just because I had the prerogative to do so. In the case of irradiation, like you took gamma ray in a package, and you irradiate it and you kill all the stuff, doesn't that leave a residue of material left that you kill the bugs, but they still have bodies? So what's
Exactly.
And it's in a package closed. So
Exactly.
what's the criteria for dealing with that?
Yeah.
That too has a minimum or maximum.
Absolutely. And that actually, so I'm glad you asked that question because that relates, and that's the cell wall of the bacteria, right? The lipopolysaccharide component of the bacteria, and that's picked up by endotoxin testing.
Okay.
So we use endotoxin testing to evaluate that. So even if you have, destroyed, beat up little micro bodies covering your device.
I mean, you wouldn't do that in an actual delivered device, but you'd have to take one out, sample it, and do that, and then
Exactly.
then make the assumption that it correlates to all the others.
Exactly, and typically they do a sampling plan where they'll say, okay, we produced 1,000 of these devices, and they're going to send us. So there's certain statistical methods or sampling plans that they utilize to determine, hey, we need to send them 60 devices, and all 60 devices need to pass the acceptance criteria for us to.
Because you can scale with concentration, because
Yep.
if they're down and if they're a high concentration, the more bodies, but still it goes in there. But all that's worked out, right? A lot of statistical analysis is in there, that's buried in your. Let's thank John again.
Yeah.
Right.
Yeah, that's great.
Thanks, John.
Hey, thank you.
Very appreciative. Thank you.